Hello everyone, my name is Novia Mann. I'm a second-year graduate student at the Virginia Institute of Marine Science, and today I'll be giving a brief update on the EC2 project that my advisor, Dr. Song, and I have been working on along with our collaborators. So far, our project has been aimed at assessing how microbial communities change as we move along the terrestrial aquatic interface of wetland ecosystems within the Chesapeake Bay. So wetlands are these highly productive habitats that provide many important ecosystem services, such as the roles they play in carbon cycling and organic matter sequestration. The microbial activity within these ecosystems directly contribute to their ability to carry out such valuable services. This is so as microbes play an essential role in wetland carbon cycling through the mediation of important biogeochemical processes. However, wetlands are quite dynamic. They're typically subjected to significant hydrological, ecological, and biogeochemical variability over relatively small spatial scales. So in order to get a complete understanding of the state and function of these ecosystems across their terrestrial aquatic interfaces, it's important for us to understand how these microbial communities are changing across this interface as well. With that said, our main objective for this project has been to firstly evaluate how microbial communities vary across distinct geochemical gradients. This gradient included soil samples from the coastal upland forest through their transitional forest all the way to the nearshore sediments. Our second objective has been to evaluate how microbial communities vary from one wetland habitat to another along the salinity gradient within the Chesapeake Bay. In order to do this, our collaborators sent us a total of 57 samples from wetland sites across the Chesapeake Bay. And while we're still working on sequencing DNA from all 57 samples, earlier this summer we conducted a pilot study with 14 of these 57 samples. These 14 samples came from 4 wetland sites in the York River estuary and are all of different salinities. They are Sweet Hall Marsh, which is our tidal freshwater wetland, Tascanis Creek and Catlet Island, which are our brackish wetland sites, and Goodwill Island, which is our salt marsh site. The samples used in this study were collected in fall of 2021 and sent to us earlier this year. Once we received the samples, we extracted the environmental DNA from each using the DNeasy Paralyzer Parasoil Pro Kit. Then we amplified the DNA using barcoded 515 forward primers and 806 reverse primers, and this was done to target the V4 regions of the 16S rRNA genes. Once our DNA was amplified and purified, we then sequenced that DNA using Illumina's MiSeq platform. These sequences were processed using the Data2 open source software. We then used the Silva reference database to match taxonomic information to the sequences. And then various RStudio packages were used for all graphical and statistical analysis. And these are the results of this analysis. Here we're looking at the principal coordinate analysis for the whole microbial community from each of our 14 samples. And essentially, this figure tells us how similar the microbial communities of each samples are to each other. The closer the points are together, the more similar those communities are, and the further away, the more different. The various colors highlight samples from different wetlands, and the various symbols indicate where the samples came from within that wetland. While overall, the microbial communities are fairly different from each other, we can see that there are some similarities among positions along a transect. For example, the upland samples are fairly close to each other, and these two wetland samples are also fairly close to each other. This tells us that the microbial communities from similar points along the geochemical gradient are more likely to be similar in structure and function than those from the same wetland. Next, let's look at the microbial composition of all of our samples. And here we're looking at the microbial community from the phylum taxonomic level. And as it's a little difficult to see what's happening with all 4 wetlands on the same figure, let us first just focus on one wetland at a time. This way, we can better see changes that are occurring along the transect. And just to get us orientated with this figure, this first bar here represents our sediment sample, the next, our wetland sample, followed by our transitional forest sample, and lastly, we have our upland forest sample. And right away, we can see that there are a few trends that are occurring along the transect. These are most likely induced by gradual changes in hydrology. For example, we have an increase in actinobacteriota as we move from the sediment to the upland. So these are a group of organisms that can live in both aquatic and terrestrial environments, but are clearly much more abundant in terrestrial habitats. Similarly, we have chloroflexi, which can be found in a diverse range of habitats, but are much more abundant in aquatic environments. I also wanted to bring to your attention the sulfobacteriota. So these are sulfate reducers. And notice that while there isn't a defined increasing or decreasing trend, there are none present in the upland sample. And if we look at changes along the transect within Tascanis Creek, we see similar trends as those found in Sweet Hall Marsh. Some communities have increasing abundances, while others have decreasing abundances. And once again, there are no sulfate reducers in the upland. And this is consistent among all upland samples. They have no sulfate reducing microbial communities present. Okay, so with the sequencing data that we collected, we can also examine microbial composition and their relative abundance at the class level. And since there were none present in the upland of our samples, here we're taking a more detailed look at the sulfate reducer community composition. So these are actually anaerobic microbes that use sulfate instead of oxygen as their terminal electron acceptor during organic matter degradation. And they're actually quite abundant in marine environments. And I mainly wanted to highlight that as we move from one wetland to the next, the diversity and relative abundance of specific groups of sulfate reducers vary quite drastically with salinity. For example, if we were to compare the sediments and wetland samples from our freshwater site, Sweet Hall Marsh, to our salt marsh site, Goodwin Island, you can see that Sweet Hall Marsh has a much more diverse group of sulfate reducers. Moving forward, I'm excited to see what this analysis will look like with samples throughout the Chesapeake Bay, and we're working on getting that data out as soon as possible. In fact, we have already extracted the environmental DNA from all 57 samples and sequenced about half of those samples. Once we have the second half of our sample sequence, we will be conducting our bioinformatic and statistical analysis. All the data that we collect from this study will be publicly available through the Exchange Public Data Repository. And lastly, I just wanted to mention that while the first campaign provided us with a lot of site-specific information, any other site-specific environmental metadata will be helpful in contextualizing this microbial community information. And with that said, I'd like to thank the large team of people who've made this project possible and all the various institutions that have contributed to this effort as well.